This is me Arzu here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 2 video of the periodontal ligament, and in this video we will be discussing with the components of the periodontal ligament. In this video we are going to discuss with the different kinds of the cells, okay? So the different kinds of cells that are present in the periodontal ligament, and also there is extracellular substance in this periodontal ligament, and about this extracellular substance, fibers and the ground substance, okay, we will be discussing in the next video, okay? As this cell portion is also one of the important subtopic of this chapter, so I thought to discuss this all cells properly. Previously also in the cementum video we have discussed with the some of the cells. We have discussed with I think fibroblast, okay, cementoblast we have discussed there, but properly we will discuss today, okay? And in this video I have prepared a proper table, okay, that I have taken from the Orbans. That table is also present in Orbans and I will be discussing with that table itself, okay? So in the previous video I think you have watched that part 1 video of the periodontal ligament. There I have discussed properly about the periodontal ligament, okay? So that was the introductory video of this periodontal ligament, and I have discussed the different kinds of theories also there, okay? So 3 theories were present there. I think you have completed that video and you are watching this, okay? So without delaying much, let's get started. Let's start our part 2. So okay, so as I have told, we will discuss with the periodontal ligament components, okay? So the composition of the periodontal ligament in this part 2 video, okay? So firstly we should know that periodontal ligament have the cell and the extracellular substance, okay? So in any histological thing, okay, in any histological substances we have particularly cells and the extracellular substance present there, okay? So extracellular substance means the space or the substance that is present between the cells, okay? So it is a space that is present between the cells, okay? So in cell also we have different kinds of cells, okay? So we have particularly 5 different types of the cell. So first type is synthetic cell, another is resorptive cell, another is progenitor cells, epithelial rest of molasses, and defense cells, okay? So in the synthetic cell we have 3 other different types of the cells. So one is osteoblast, another is a fibroblast, cementoblast. So blast, okay? So this blast means synthetic, okay? So it do synthesis of proteins, it do synthesis of the cells, okay, protein substances, okay? So it is synthetic cells. So blast means synthetic cell, osteoblast, fibroblast, cementoblast, and we will discuss with it properly. And in resorptive cell we have clast, so osteoclast, cementoclast, and in fibroblast we can see it is not written fibroclast, okay? So there is no any kind of fibroclast cells, okay? So the resorptive cell of resorptive cell is also, so this fibroblast is the cell, okay? So it do synthesis also and also do remodeling, okay? I will discuss with the fibroblast properly. So in resorptive cell also we take fibroblast itself, okay? There is no any kind of other type of cell that is named fibroclast. There is no any such cell, okay? So it is a fibroblast which do synthesis also and also do resorption. So we have taken fibroblast here, and another is a progenitor cell, stem cells, okay? And another is epithelial list of molasses, and we have discussed with this epithelial list of molasses properly in the development of this video, okay? Properly we have discussed with this, okay? And after that in the defense cell we have mast cell, macrophage, and eosinophil, and we have also discussed with this defense cell in our pulp chapter. In part 3 video of the pulp we have discussed little bit, and also in this periodontal ligament, as it is a subtopic of periodontal ligament, we will discuss with this defense cells. And in the extracellular substance we have particularly fiber and the ground substance, okay? So this fiber means collagen fiber, elastic oxytocin fibers are present there, reticular fiber, secondary fiber, in different fiber plexus, and we will discuss with all those fiber, okay? So there are principal fibers, okay, in this periodontal ligament. We will be discussing with all those fiber in upcoming videos. And about this ground substance, okay? So ground substance we have glycosamine glycans, proteoglycans, and glycoprotein present in the ground substance of the periodontal ligament, okay? So we have discussed with just the overviews, and this flow chart is one of the very important flow chart, okay? So they can ask you in the theory exam, so draw so write the composition of the periodontal ligament. So in such a case you can draw this flow chart. Okay, so just remember there is cell and the extracellular substance. In cell we have 5 different types of the cell: synthetic, resorptive, progenitor, epithelial, of molasses, and defense cell. And in extracellular substance we have fiber and the ground substance. In ground substance there is glycosamine glycan, proteoglycans, and glycoprotein. And the fiber we have other 5 different types of the fiber. Okay, okay. So now in another paper, so as you can see here I have prepared a proper table. Okay, so proper table I have prepared, and this table is taken from the Orban. So I will be discussing every point, okay, that is present in Orban, so you shouldn't have to be worried. Okay, so we will discuss with the uh, osteoblast. Okay, so osteoblast is a synthetic cell. So osteoblast, it lines the bone surface and bone forming cell. Okay, so about this osteoblast I think you have discussed in the general histology. Okay, so of general anatomy, so there you have been discussed with this osteoblast. Okay, so osteoblast are the bone forming cell and they just line the bone surface. After that, the origin of the osteoblast is primitive mesenchymal stem cell. Okay, so it originates from the primitive mesenchymal stem cells. Okay, and in the light microscope and in the electron microscope. Okay, so for the morphology of this osteoblast we will discuss light microscopy and electron microscopy of every cells. Okay, so in every cell we will discuss with this light microscopy and the electron microscopy, how they appear in a light microscope and electron microscope. Okay, so in the light microscope, so osteoblasts are seen cuboidal. Okay, so they are seen cuboidal in shape with a round nucleus at the basal end of the cell. Okay, so they are the cuboidal cells, and round nucleus is present at the base side. Okay, so at the base end, at the lower third, okay, so there is a presence of the nucleus that is present in this light microscope. Okay, and in electron microscope we can see abundant rough endoplasmic reticulum, Golgi complex, mitochondria, and vesicles are present there. Okay, so abundant number of rough endoplasmic reticulum, Golgi complex, mitochondria, vesicles, and it is basophilic cell. Okay, so the basophilic, yeah, staining take place in this osteoblast. Okay, okay, so you can remember this by a proper concept. Okay, so this osteoblast is a synthetic cell, means it synthesizes the protein. Different kind of protein are synthesized by the osteoblast. So there must be different organelles, okay, that is responsible for the protein synthesis. And this rough endoplasmic reticulum, Golgi complex, and mitochondria, vesicles, they are those organelles. Okay, so uh, these are those components of the cells that are particularly responsible with the protein synthesis. So those components should be in higher number, this should be abundant, and that is a concept. Okay, so and it is similar in this fibroblast also, and also in the cementoblast, as all three are the synthetic cell. Okay, but will be different in the resorptive, so that we'll discuss now. And the functions, okay, so we have 4 particular function. And function number 1 is a formation of the new bone, as it is a bone forming cell, regulation of the bone remodeling. Okay, so also osteoblast, it also regulates the bone remodeling. Okay, and mineralization of the osteoid and the secretion of the collagenous and non-collagenous protein. So it is a synthesizing cell, it secretes the protein, so it secretes the collagenous and the non-collagenous protein, type 5 collagen, osteonectin, rancal. Okay, different kinds of the collagenous and the non-collagenous protein are just secreted. Okay, so we have completed with the osteoblast. So just remember, it is a bone forming cell. Okay, lines the bone surface. Primitive mesenchymal stem cell is it is its origin. And in light microscope it is cuboidal with a basal end having the nucleus. In electron microscope it is abundant, it have abundant rough endoplasmic Golgi complex, and vesicles are present there that are responsible for the protein synthesis, and we have discussed with the functions. Okay, okay, so now another, our cell is the fibroblast, and it is one of the prominent cell that is present in the periodontal ligament, and that is present 35% of the whole cell that is present in the periodontal ligament. Okay, okay, so I have written a small point here, okay, about this gingival fibroblast. Okay, so we have particularly 2 different kinds of the fibroblast. One is gingival fibroblast and another is a periodontal ligament fibroblast. Okay, so gingival fibroblast means the fibroblast that is present in the gingiva, and in periodontal ligament those fibroblasts that are present, that are called periodontal fibroblast. So there is some little difference, okay, but the morphology are quite similar, but there is little difference in this gingival fibroblast and the periodontal fibroblast. So that is, so it is mesodermal in origin. Okay, so it is mesodermal in origin. Similarly, as the this periodontal ligament fibroblast, it is a more synthesizing cell. Okay, it just secrete the more protein, but these gingival fibroblasts, they are not more synthesizing cell, so there is a less number of ALP. Okay, so less ALP, alkaline phosphatase, and cyclic AMP is also less there. So the proliferative capacity, okay, so these are related with the proliferation of the cell. So this ALP and the cyclic AMP is less in this gingival fibroblast, so there is a less in a proliferative capacity, and there is no any cytoskeleton present in this gingival fibroblast. Okay, so we have discussed with the gingival fibroblast. So just you should remember is that, okay, so they are mesodermal in origin and they have less proliferative capacity, so they have less number of ALPs and cyclic AMP, and there is no any cytoskeleton present. Actin and actin filament, okay, so there is absent in this gingival fibroblast, but that is present in this periodontal ligament fibroblast. Okay, so this is one of the important point you should remember. Okay, so the cytoskeleton that is present, I have also written here about the cytoskeleton. So the cytoskeleton that is present in this periodontal ligament fibroblast, it is very important. Okay, it have greater importance in the tooth eruption. So there is a periodontal ligament traction theory in the tooth eruption. Okay, so the actin, so this periodontal ligament fibroblast have the contractile properties. They have the cytoskeleton, actin filaments are present there, and those filaments are responsible for the tooth eruption, and that I think I have discussed previously. Okay, so in the function video, in the first video of this periodontal ligament. Okay, okay, so in this origin of this fibroblast, okay, you just have to remember. Okay, so as I have written in the first one, so there I have written from partly from the ectomesenchyme of the investing layer of the dental papillae and the dental follicle, and this is one of the important point you should remember in origin. Okay, so here are further 2 points. Okay, so I have written about to this fibroblast. Okay, so those fibroblasts that are near to the cementum. Okay, so the fibroblasts that are near to the cementum, they are from this ectomesenchymal, so it is similar with this point, and the fibroblast that is far away, so that is near to the bone and far away from the cementum, they are from the perivascular mesenchyme. Okay, so you should have to remember that fibroblast near the alveolar bone, it forms from the perivascular mesenchyme, but near cementum it forms from the ectomesenchymal cell of the investing layer. Okay, so you should remember this ectomesenchymal cell of the investing layer of the dental papillae and the dental follicle. Okay, so that is the proper origin of the fibroblast. Now in the light microscopy, okay, so we have active fibroblast, another is a resting fibroblast. Okay, we have active fibroblast, okay, and another is a resting fibroblast. So in the active fibroblast, it is rich in a cytoplasm, oval nucleus, and one or more prominent nuclei is present there. Okay, after that it is fusiform or the tripolar with the fine cytoplasmic process, or the stellate with the numerous short processes. Okay, so it is a fusiform or the tripolar with the long fine cytoplasmic processes, or stellate with the numerous short processes. And the resting fibroblast, it is a flattened dark staining nucleus is there, and it have little cytoplasm. Okay, so particularly the fibroblasts are related with the protein synthesis. Okay, so there must be cytoplasm that is rich, and nucleus is also there with one or two prominent nuclei. Also there is a fusiform or the tripolar. The fibroblast, they appear fusiform or the tripolar with a long fine cytoplasmic processes are also there. Okay, so they are like this. Okay, so this is a cytoplasmic process. Okay, so long fine cytoplasmic processes are there. Okay, so that is present in this fibroblast. After that, in the resting fibroblast, it is just a opposite of that active fibroblast. So there is a less number of the cytoplasm, and there is a dark staining nucleus present there. And in the electron microscope, we can see a bonding pair of endoplasmic reticulum, which is similar with that of the osteoblast. Golgi complex is more, mitochondria is more, secretive vesicles is more, and well-developed cytoskeleton is also present there, which consists of the microtubule and the microfilament. Okay, so we have completed with the morphology of fibroblast, and in the function section, we can see it secretes the collagen, elastin, structural connective tissue protein. Okay, so it secretes the collagen and the elastin, and those are the structural connective tissue protein. Okay, which they are the particularly structural proteins. And another point secretes the proteoglycans, glycoprotein and the GAGs, okay, and those are the ground substance, okay. So those are the ground substance of the periodontal ligament. And after that, in the next one, matrix metalloproteinases. So this is an enzyme that is secreted by this fibroblast, and this is one kind of MCQ also. So they can frame it in a question, okay, asking that matrix metalloproteinases are secreted from the which cell of the periodontal ligament. Then you should tell fibroblast is a cell that secretes this matrix metalloproteinases, and in short form we call it MMP, okay, MMP. After that, in fourth point, remodeling of the periodontal ligament fiber, okay, and maintains the width of the periodontal ligament and thickness of it, okay. So width, so this is fibroblast, okay. So I have discussed with those point in first video. There I have in periodontal ligament homeostasis, okay. So there I have discussed particularly with those width, okay, thickness of the periodontal ligament, how it affect the periodontal ligament, okay. So those comes under the homeostasis section, okay, and I have discussed with those. And also it is the growth factor cytokines, okay. So by those growth factors and the cytokines, they just maintain the homeostasis of the periodontal ligament. They maintain the width of the periodontal ligament and the thickness of it, okay. So we have completed with the fibroblast, and this is one of the important cells, okay. You should remember about the origin. You should just remember ectomesenchymal cell of the investing layer of the dental papillae or the dental follicle, and near alveolar bone it by perivascular mesenchyme. So these are the origin point of this fibroblast. And there is also another fibroblast, gingival fibroblast, that is mesodermal in origin, and it have less proliferative capacity. So it have less ALP and the cyclic AMP, and also there is no cytoskeleton like that of the periodontal ligament fibroblast, okay. After that, in a third cells, okay, so in third cells we have cementoblast, okay. So in cementoblast we can see line surface of the cementum, okay. So as it is a cementum forming cell, okay, so this is particularly cementum forming cell. So it lines the surface of the cementum, and it is developed from the dental follicle and the hot epithelial root sheath, okay. So about this cementum, okay. So in the dental development of the tooth video I have discussed properly, okay. So which part of the tooth is developed from the which portion of the enamel organ. So there I have discussed that dental follicle forms the cementum, okay. So dental follicle forms the cementoblast, and cementoblast forms the cementum, okay. So now in the light microscope we can see the cubital cells with the abundant cytoplasm and a large vesicular nucleus with one or more nuclei, okay. So this is also a cubital, that is a bit similar with that of the osteoblast, and abundant cytoplasm is there, and large vesicular nucleus is also present there in cementoblast. After that, in electron microscopy we can see that all proteins releasing organelles are present here, okay. Okay, so in this electron microscopy, okay, so the all protein releasing organelles are present there, and cells depositing cellular cementum have the abundant cytoplasmic processes, abundant mitochondria, and but less rough endoplasmic reticulum, okay. So this is important. So there was a more number of rough endoplasmic reticulum in the osteoblast and the fibroblast, but there is less number of RER in this cementoblast. Other or are quite similar, okay. And in the function section it lays down the cementum that helps in the attachment of the tooth to the alveolar bone via the collagen fiber of the periodontal ligament, okay. So it just forms the cementum, okay. Now we will move to the resorptive cell, okay. And in the resorptive cell, first we will discuss with the osteoclast. And this osteoclast, the origin of the osteoclast is from the hematopoietic stem cells of the monocyte macrophage lineage, okay. So this is the origin of this osteoclast. And in the light microscopy it is large and multinucleated, okay. So it is large and multinucleated, and it is eosinophilic, okay. And it have eosinophilic cytoplasm, but it was quite a basophilic in the osteoblast and the fibroblast, okay. But it is eosinophilic in this osteoclast. After that, cells found within a resorptive lacunae, that is called Hausip lacunae, and this is very important, okay. So this osteoclast are present, and the cementoclast are also present in this Hausip lacunae. So now in this electron microscopy, okay, so we can see numerous mitochondria and the lysosome, okay. So these lysosomes are mostly present in this clast type of cell, and abundant Golgi sacules and the free ribosomes, but little RER, okay. but little area. So that is the morphology of this osteoclast in the electron microscope. After that, in the function section, it plays an important role in the bone resorption, also responsible for the remodeling. Okay, so we have completed this osteoclast, and after that, in a cementoclast, we can see the origin. Okay, so cementoclast means resorpting the destroying the uh, it do resorption of the cementum. Okay, so origin is the unknown. Okay, so the origin is particularly unknown of the cementoclast, but arise in the same manner as that of the osteoclast. Okay, so in some of the manner, some of the researcher told that used to tell that cementoclast is particularly developed from the hematopoietic stem cell of the monocyte macrophage lineage. Okay, so that is also the origin of the osteoclast. After that, in the light microscopy, the cementoclast. Okay, so they are mononuclear or the multinucleated giant cell in the house lacunae of the cementum. So the morphology is bit similar that of the osteoclast. Okay, so in osteoclast also there was the large and the multinucleated cell present with the eosinophilic cytoplasm, and it is present in the house lacuna, and this cementoclast is also similar with that. Okay, and giant cell means larger cell. Okay, so there was also a giant cell, there was also a larger, and it is similar with that of the osteoclast, and both this osteoclast and cementoclast are bit similar. Okay, every point. So the origin also similar, light microscope is also similar, electron microscope is also similar, and the function is also similar. So resorption, and this osteoclast also do the resorption, okay, of the bone. Okay, so about osteoclast and about the cementoclast we have discussed, that comes under the resorptive cell. And about the fibroblast we have particularly discussed previously itself in the synthetic cell portion. Okay, so now we will move to the another type of the cell, that is epithelial rest of molasses. Okay, so that is, this is another type of the cell that is present in the periodontal ligament, and it is abundant on the forcation area. So this can be framed into the MCQ. So epithelial rest of molasses are mostly present at the forcation areas. Okay, forcation areas means it is between the two roots, okay, of the posterior tooth. Okay, so that is present in the forcation areas, and it is parallel to the root surfaces. After that, the origin. Okay, during the time of the cementum formation, epithelium that covers the surface of the newly formed dentin breaks into the lace-like strand. Okay, and okay, so this epithelial list of molasses, they are particularly formed, okay, during the time of cementum formation. Okay, so when the cementum is formed, so there is a epithelium that covers the surface of the newly formed dentin. Okay, so there is a epithelium that covers the newly formed dentin, and those epithelium breaks into the laces. Okay, so they just breaks into the laces like a strand, and those cells, those laces of strands, okay, just give this epithelial list of molasses, and this is a remnant. Okay, so I have about discussed about this epithelial list of molasses in the development of the tooth video properly, so I will not be discussing it more here. After that, in the light microscopy, okay, so the cross section cluster like, so it appears cluster like in the cross section, and in the tangential section, network of interconnecting strand parallel to the long axis of the root we can see. After that, in the electron microscopy, we can see the scanty cytoplasm with the tonofibrils that insert into the desmosomes between the adjacent cell, and hemidesmosome between the cell and the basal lamina. Okay, so there is a scant cytoplasm with the tonofibrils. Okay, so there is a tonofibrils that inserts into the desmosomes, and or the hemidesmosomes. Okay, after that also I have written more about this electron microscopy. So the mitochondria is distributed throughout the cytoplasm. Okay, and RER Golgi apparatus are poorly developed in this epithelial list of molasses. Okay, and also this is a remnant of the Hartwig epithelial root sheath. Okay, so that I have told, I think so it is one of the remnant. Okay, and it is a remnant of the Hartwig epithelial root sheath, and it is lie about 25 micrometer from the cementum surface. Okay, so it lie about 25 micrometer from the cementum surface. After that, so I have written here about, so particularly the functions of this epithelial rest of molasses is not known. Okay, but we have clinical significance of this epithelial rest of molasses, and also the distribution. Okay, so in this clinical significance, so we can see here the proliferation to form cyst and the tumor, or may also undergo the calcification forming the cementicles. Okay, so it is a bit related with that of the pathology. Okay, and also I have told in the development of the tooth video, so they are particularly developed into the cyst. Okay, so they just develop into the cyst. Okay, so they just proliferate and they form the cyst or the tumors that undergo the calcification, and so on the cementicles. Okay, so cementicles are formed there. After that, in the distribution, so it is less numerous in the older but more numerous in the children, and in second decade in apical region. Okay, so in apical region it is mostly present in the second decade. After that, later cervically in the gingiva above the alveolar crest. Okay, so alveolar crest means, so this is alveolar bone, so this is a crest. Okay, so the topmost portion of the alveolar bone is the crest, and above this there is a gingiva. Okay, and there is a tissue. Okay, so it is present here. Okay, later in life, so cervically in the gingiva above the alveolar crest. After that, so we have discussed with this distribution also of this epithelial list of molasses. Now we will discuss with the progenitor cells. Okay, so these progenitor cells are mesenchymal stem cell responsible for the tissue homeostasis. Okay, so they are responsible for the tissue homeostasis, and it is originated from the undifferentiated mesenchymal cells that are pluripotent. Okay, so they are particularly the stem cells. Okay, they can form any other type of the cells, just like fibroblast, osteoblast, osteoclast, fibroclast. Okay, so it just maintain the homeostasis of the periodontal ligament. It just maintain the quantity of the cell that is required at the moment in the periodontal ligament. So they are the stem cell particularly, and cell have the prominent nucleus and the little cytoplasm with a perivascular location. Okay, so they are particularly present around the blood vessels. Now the functions: sustain, renewal, and the regeneration. Okay, so that is a proper function of this stem cell, progenitor cell. Okay, so you guys should just remember progenitor cells are particularly the stem cell, and they are formed from the undifferentiated mesenchymal cells which are pluripotent. Okay, so they are derived from this undifferentiated mesenchymal cell, and they are responsible for the sustain, renewal, and the regeneration. Okay, after that, in the eighth, our eighth cell is a mast cell. Okay, so this comes under the defense category. So the mast cell are associated with the blood vessels, so they are particularly associated with the blood vessels itself. Okay, and the origin is the hematopoietic stem cell. Okay, and here is also hematopoietic stem cell, and here also you can see the hematopoietic stem cell. So you should remember all the defense cells that are present in the periodontal ligament are particularly derived from the hematopoietic stem cells. Okay, so now in the light microscopy, as I have written here, so it is round or oval cell 12 to 15 micrometer in the diameter. Okay, so it is round or the oval cell with the 12 to 15 micrometer in the diameter, and the numerous cytoplasmic granules, okay, are present here. Numerous cytoplasmic granules are present in this mast cell, and those granules, okay, so they are the membrane bound vesicles or the granules that is of 0.5 to 1 micrometer in diameter, and they contain the heparin and the histamine, and they are stained with the metachromatic dyes. Okay, so you should just remember mast cell. Okay, so they are round or oval in shape, and they are 12 to 15 micrometer in the diameter with and also it contains different kinds of the metachromatic granules. Okay, so there are the granules, cytoplasmic granules are present there, and those granules are 0.5 to 1 micrometer in the diameter, and those granules contains the heparin and the histamines. Okay, so those chemical substances are present in those granules. After that, in the electron microscope, we can see cytoplasm and free ribosomes are present there. Short profiles of the granular endoplasmic reticulum is also there, and a few round mitochondria and prominent Golgi apparatus we can see in the mast cell. And the function of the mast cell is that it releases the histamine and into the extracellular environment, proliferation of the endothelial cell and the mesenchymal cell. Okay, so that is a particular function of the mast cell. Okay, so it is a defense cell. Okay, so it just releases this histamine and the heparin. Okay, and that just into those extracellular environment, and they just proliferate the endothelial cell and the mesenchymal cell. Okay, so they are particularly related with that of the defense cell. Okay, and I think you have discussed with this mast cell in detail in the general pathology, so I won't discuss it here more. Okay, and it is also not bit important in this periodontal ligament. After that, last step, we have macrophages. Okay, and macrophages, they are also located adjacent to the blood vessel, similar with that of the mast cell. And hematopoietic stem cell is its origin. And the in light microscope, we can see the nucleus, that is horseshoe shaped or kidney shaped with the dense and uneven layer of the peripheral chromatin. Okay, so nucleus that is present here, they are particularly the horse shaped or the kidney shaped with the dense and uneven layer of the peripheral chromatin, and nucleoli are rarely seen in this macrophages. Okay, so nucleoli are rarely seen here, and you just have to remember the shape of the nucleus. Okay, and in the electron microscope, we have resting macrophages distinguished from the fibroblast by the presence of the numerous microvilli. Lysosomes are present here, and membrane bound vesicles, okay, of varying density and porosity of the rough endoplasmic reticulum and the Golgi complex. Okay, so in the macrophages located, that is adjacent to the blood vessel. Okay, so they have the nucleus, and the nucleus that is a horseshoe shape or the kidney shape, and in electron microscope we can just see the numerous microvilli, numerous lysosomes, membrane bound vesicles are present there that are responsible for the degradation of the foreign particle. Okay, and in the macrophage they have dual role. Okay, and phagocyte dead cells secretes the growth factor, regulates the proliferation of the adjacent fibroblast, and also synthesize the interferon, prostaglandins, and the factors that are enhancing the growth of the fibroblast and the endothelial cell. Okay, so all those things I think you know well about this macrophages. So they just engulf the foreign particle and they just degrade those foreign particle. Okay, phagocytose, those dead cell, also secretes the growth factor and regulates the proliferation of this fibroblast. After that, our last cell, eosinophil. Okay, so they are occasionally seen in this periodontal ligament, and the origin is also similar with that of the macrophage and the mast cell. So that is a hematopoietic stem cell, and in light microscopy we can see 8 micrometer diameter nuclei are bilobed granules with one or more crystalloid structure with bright red staining properties with acidic dyes such as eosin. Okay, so that is in the light microscopy. Okay, so in the eosinophil we can just see 8 micrometer diameter cell. Okay, and that is a eosinophil, and nuclei is bilobed. Okay, so there is a bilobed presence in this nuclei with the granules with one or more crystalloid structure are present there, and brighter staining properties with the acidic dyes such as eosin. So it have brighter staining properties. Okay, so there is also a granule similar with that of the macrophage, and the function is the phagocytosis. Okay, and I won't discuss more about this eosinophil. Okay, you can just remember, and this is not bit important in this chapter too. Okay, so we have completed with this defense cells. Okay, synthetic cell, for resorptive cell, epithelial layer of molasses, and the one cell progenitor cell. Okay, so we have completed all those cells that are present in the periodontal ligament, and in the next video we will discuss with the extracellular substances. There we will discuss with the fibers, and that is also one of the important subtopic of this chapter. Okay, so in that video we will discuss with different kinds of the fiber. Okay, and that is been asked in the theory exams or in the viva exams, so we will discuss that in our next video. So for today we have completed our topics. Okay, so I will meet you in the next video. Till then, stay happy, stay healthy, and tata, bye bye, thank you. And also if you need the notes of this video, okay, so you can visit my telegram channel, and the link of the telegram channel is in the description below. Okay, so in the description of this video you can get the link of the telegram channel. So I used to upload the notes of the all the videos, okay, that I have discussed in my channel. Okay, so those notes, okay, so those all notes are present in my telegram channel. Okay, so you can visit that. Okay, so I'll meet you in the next video. Till then, stay happy, thank you.